Okay, so this is part 2 of cardiovascular, and in this one, this is actually very important, so it's probably going to take a while, just to warn you. And we're going to talk about the electrical properties of the heart, and this is actually what makes the heart very unique. It's the focus of a lot of drugs. It's the cause of a lot of heart problems. And so it's kind of important that you understand all of these things. First of all, we're going to start out talking about the autorhythmic cells. And then we'll talk about contraction and how calcium plays a big role that's very different in cardiac muscle as compared to uh, striated muscle or skeletal muscle. Um, I mean skeletal, striated, of course. Cardiac muscle is also striated. And then we'll talk about the EKG and finally the cardiac cycle. So when I did labs at the University of Kansas long, long ago, we did a lab where we took a heart from a frog, removed it completely, and put it into a solution called Ringer solution, which had all the electrolytes and everything that it needed to keep beating, and it did. It kept beating, which means that your heart, every time it beats, is not getting directions from your brain or from your spinal cord. It is all contained within the heart. So we call those autorhythmic cells. And there is a group of autorhythmic cells. There are actually several groups of autorhythmic cells that exist in your heart that depolarize on a regular basis, continuously, about, if left alone, around, 90 times a minute. Um, and that's what keeps your heart beating all of the time. Okay? So we call those autorhythmic cells, we call them pacemaker cells. Okay, and they sit in different places. This is showing the SA node. And so here are a bunch of cells that just kind of sit in here and they will depolarize. Okay, um, and and spontaneously. And the reason that they spontaneously depolarize is because they're actually leaky to sodium and calcium. But first of all, they're leaky to sodium. Okay, now that means a lot because we know that if you have if you have high blood pressure or something, you're supposed to avoid sodium. Well, this is one of the reasons you avoid sodium, because if you have more sodium, then these tend to be more leaky, or they're not more leaky, but the sodium rushes in more quickly and it causes them to depolarize more quickly, so your heart rate is increased. Okay, so you have this group of cells, and then they depolarize spontaneously, and that's what this graph is showing, is that they are sort of depolarizing, and then they have an action potential, repolarize, depolarize. So they're doing the same thing that we've learned with every other action potential. The difference is that nothing is telling them to do it. It's just because they're leaking sodium. Okay, so when it reaches its threshold, it has an action potential, just like we've always seen. When something reaches threshold, it triggers the action potential, and the sodium rushes in and the potassium rushes out. Um, but because this is just a small group of cells, what it does is it travels through, remember, gap junctions. Okay, so they have gap junctions. This is what causes all of these cells these are the intercalated discs, this is what causes all these cells to sort of act as a single unit. And so all you have to do is get depolarization in this area, okay, which this one is the SA node. There's also the AV node, the bundle of His, um, and the Purkinje fibers that we'll talk about later. Okay, and sympathetic activation and parasympathetic activation will speed it up, so it causes epinephrine or norepinephrine to be released, and that will speed it up. And then from the vagus nerve, you release acetylcholine, it will slow it down. And we actually, in this experiment, where we removed the heart, we actually dripped norepinephrine onto the heart. We could see that it increased the contraction rate, and we dripped acetylcholine onto the heart, and we actually saw that it decreased the contraction rate. Now I know, yes, it's the same acetylcholine that activates skeletal muscles, but in this case it's doing something a little different. We're not going to get into the mechanisms, but when acetylcholine is released onto these pacemaker cells, it actually causes them to depolarize more slowly. Okay, and so it slows the heart. Okay, so yeah, we don't have to know that mechanism in here, but that's what it's doing. Okay, so these autorhythmic cells are what keeps the heart beating constantly, all the time. Okay, And as I said, the rhythm comes from the heart itself. It's not from anywhere else. Not from the brain. It's not from the skeleton, the spinal cord. It's just from these pacemaker cells. And we have technically four of them. Sometimes the last two are grouped together. Um, but the first one we call the SA node, or the sinoatrial node, and it sits right here. Okay, so it sits in the upper right part of the heart. Okay, right up here in the corner. And all it is a small group of cells, and it will then cause depolarization of all of the atria. Okay, so depolarization of all the atria is what it causes. Okay. Now these are just a small group of cells that's automatically doing it, but then they transfer that signal to the atria. Okay, and then there's another one that's a little bit slower. So this one, if left alone, would be about 100 times per minute. Some books say 90 times per minute. And so we're controlling that constantly. We're constantly, with our sympathetic and parasympathetic nervous system, we're changing it so it's not stuck on that number, obviously. In fact, it's usually a lot lower than that. But if just without any other outside influence, that's how often it would depolarize. The second pacemaker cell, or group of cells, is called the AV node, or the atrioventricular. Atrioventricular, where do you think it sits? Right between the atria and the ventricles. It sits right here, okay? On the atrial side, but it's right down there between the atria and the ventricles, okay? So that's where it is, right there. Okay? And it will, if left alone, depolarize at about 40 to 60 beats a minute. Now, the significance of this is that a lot of people get something like called sick sinus syndrome. And what that is that's a condition where your SA node will fail. And a lot of times the SA node fails for a variety of reasons. Heart disease, which is a vague term, but it will just fail. And some people don't know why it fails. But what will happen is if this one doesn't work, well then the AV node will actually take over. Now that's not a good thing when you're used to your heart beating at about 75 or 80 beats per minute, and then the one that takes over is only beating at about 40 to 50 beats per minute. That can be a problem. Your blood pressure will drop, and you'll probably feel lightheaded or pass out even. Okay. So this is a serious problem. It doesn't. I mean, a lot of times people will have a skipped heartbeat or something like that. That's a completely different issue. But when your SA node actually fails, you do have this backup, but it's much slower. And while it will keep you alive, it may not keep you conscious. Okay, so that's the first one, the SA node, and you will be asked that very question: what is your primary pacemaker? And it's the SA node. The second one is the AV node, and then we have the third and the fourth, and I put them as third and fourth because some books will separate these and call this the bundle of His, number 3, and the Purkinje fibers, number 4. Some people just say the third one is the bundle of His and the Purkinje fiber, and here's why. Because it's not an actual node. These guys aren't actual nodes, but they do have autorhythmic capabilities. Okay, so I'm going to change this to black so maybe we can see it a little better. They do have autorhythmic capabilities. So here's the AV bundle right here. It's called the bundle of His, or the AV bundle, and yes, that's pronounced His. It's named after a person. It's not his. The bundle of His, AV bundle, same thing. And so these will spontaneously depolarize. So going back to this experiment we did with the frog heart, what we actually did at one point is we actually cut the atria and we separated the atria from the ventricles. And guess what? They kept beating, both of them, even though they were in completely different parts of the dish. They still kept beating. And it's not because they had any kind of communication with each other. It's because the SA node and the AV node are contained up here, and the AV bundle and the Purkinje fibers, which we haven't talked about, but the Purkinje fibers sit down here. Those were in the ventricles. So they both kept beating, but the Purkinje fibers and the AV bundle made this one beat much more slowly. So if you look here, The fourth pacemaker, we'll just go ahead and say that, or the third pacemaker, I'm sorry, the third pacemaker is the AV bundle, and it depolarizes about 20 to 30 beats per minute. Okay, and the Purkinje fibers are similar to that, maybe a little slower, but they kind of work together because if the AV bundle, you can kind of see this here, if the AV bundle depolarizes, it has direct connections with, so there's the left and right bundle branches, and it will directly come here and it will cause all of these fibers to also depolarize. Okay, so they have conducting um, tracks that will send this signal through the entire system. Okay, so if we look at what actually happens during a normal heartbeat, the AV node or the SA node depolarizes, and this is the one that's probably running you right now, unless you have a pacemaker or something like this. This is the one that has to be going all the time, or an artificial pacemaker. This is the one that has to be going all the time. And as soon as it depolarizes, it will send signals out over here to the rest of the atria, and then all of the atria depolarize at the same time. But it also sends a signal down here to the AV node, telling the AV node, I got this, I depolarized. And what that does is it triggers a pause. Okay, so you have about a part of a second pause, and this will then wait, and then it will send the signal back down through the ventricles. Now remember earlier I said all of the atria depolarize at the same time, and then it pauses, and then all of the ventricles depolarize at the same time, all the ventricle myocardia, or all the muscle cells of the ventricle and atria, depolarize at the same time. It's because the signal can only travel to get from the atria to the ventricles. The only way that it can get there is through this area right here. Okay, there's no other way for this signal, for the ventricles to know that the atria have depolarized except right through here. Okay, and so there's kind of this master switch and this built-in pause. It has slower depolarizing cells that exist in here, and so you have this this depolarization, pause, and then it moves down into here, and you have depolarization, okay, of the ventricles. Okay, so I hope that makes sense, because it's very, very important for proper functioning of the heart. That, and this is where it's going to come back to us later, where you have to have conduction through this area. If you don't get conduction through this area, then these guys are independent of one another. Okay, and we see that with a heart attack. A lot of times during a heart attack, this area, you might get a loss of blood flow on the outside of this area, and you may damage some of these cells a little bit. And so it may either slow conduction through this area, which means that your atria will depolarize, and then there will be a long pause before the ventricles depolarize. Okay, so this is an EKG, and we haven't talked about it yet, but that's what that's showing. It's showing the depolarization of the atria and the ventricles. Okay, so if you have damage here, then it will pause. So those are the third and fourth pacemakers: the AV bundle, or bundle of His, and the Purkinje fibers. Okay, so it moves down in this direction, and then it squeezes this way. Okay, so hopefully that makes sense. All right, so we've kind of mentioned some of this, the regulation of the heart rate. Remember, we're talking about mainly the SA node, okay, but it also controls the AV node, okay, because you don't want to slow down the SA node or speed up the SA node without letting the AV node know as well. And so when your autonomic nervous system, or your sympathetic nervous system, releases epinephrine or norepinephrine onto the SA node, that will speed up the heart. Okay, and now it also sends it to the AV node to let the AV node know what's going on, so it keeps them in sync, so the AV node doesn't go faster or too much slower. So it's still lagging behind, but it's trying to stay on the same pace to work as that backup. Okay, acetylcholine, same thing. It will slow down the SA node, and it will also at the same time slow down the AV node, so the AV node doesn't pass it up, doesn't pass up the SA node. We always want the AV node to be a little bit behind the SA node in terms of its rate of depolarization, auto depolarization. Okay, now we mentioned sodium increases heart rate. Now that should make sense because if the SA and AV node and all these other pacemaker cells are permeable, are leaking sodium, that more sodium, so if that's one side and then there's your little channel where it's leaking through, if you have more sodium here, then you just get more leaking through. And so it depolarizes a little bit more quickly. And same with calcium. It's also a little bit leaky. There's actually another channel that's also leaky to calcium, which can cause that to happen faster. Okay, so that's why elevated calcium, elevated sodium will increase heart rate. Now, everything happens at a higher temperature, so actually increased temperature will cause the SA node to depolarize more quickly, because remember, temperature is just the moving around of these molecules, or these atoms, and so more movement means that they're going to have more of an opportunity to move into this area. Okay, so if these guys are moving around more rapidly, then more of them are going to have a chance to move through. Okay, which works nicely because if you have an elevated, if you're running or something and your body temperature starts to increase, your heart will pump more, which will pump more to your periphery, which will actually help cool you. So there are actually beneficial reasons to have your heart beat a little bit faster when you're warm. And a lot of times that's just to help cool your body by putting that blood out on your periphery to help cool you. Okay, so a couple of terms, and these are just terms you have to know. Tachycardia is an elevated heart rate, or above 100 beats per minute, and bradycardia is a low heart rate, which is below 60 beats per minute. Okay, so you just need to know that, and a lot of times you'll see that. You'll hear about somebody having VTach or something like that, which is ventricular tachycardia. Okay, so that means the ventricles are depolarizing really, really fast, which is going to cause the rest of the heart to depolarize really, really fast. Okay, so if you have tachycardia or bradycardia, that's what that means, either elevated or a lower heart rate. Okay, so that's it when we talk about the autorhythmic cells. Um, let's talk about the actual muscle cells, the actual myocardia. So here's the autorhythmic cell. Okay, a little bit leaky to sodium, depolarizes all on its own. We'll just say for now that this is the SA node. Okay, and remember, it's depolarizing and then it's sending signals through the gap junctions into the myocardia. Okay, so into the heart muscle cells. Now, we're used to, remember with skeletal muscle, we had the motor neurons that came and released acetylcholine. That's what was causing the depolarization, okay, that moved through. This is different. The heart is different. The heart gets it from the autorhythmic cell. So it's just sharing. It's just moving from one to the next to the next to the next. Okay, so what happens, but it still depolarizes. These cells still depolarize in the same way. So this depolarization moves in and it opens something called an L type. L just stands for long-lasting or long-term calcium channel that causes calcium, okay, paying attention, calcium to move in to the cell, okay, calcium. Because I have right up here, cardiac muscle contraction is similar to skeletal muscle, but with important differences. And I put here calcium permeability. So obviously that's going to be a test question and obviously something you need to understand. Okay, so this calcium moves in and that causes more calcium to be released from the sarcoplasmic reticulum, and then everything else is the same as it was for skeletal muscle. It's still binding troponin, moving tropomyosin out of the way, and the myosin is moving along, walking along the actin filaments, causing the sarcomere to shorten. Same thing in that respect. And then to stop it, the calcium gets pumped back into the SR, and it can also, in this case, actually be pumped back out of the heart. Okay, so this is where it's a little bit different. It's a little bit different in that this permeability to calcium. Okay, so we basically just went down all of these things here. depolarization causes calcium to move in. Calcium depolarization and the existence and presence of calcium causes calcium to be released from the SR, and then you have the same events that take place for a contraction. And then the calcium is pumped back into the SR and pumped back out of the cardiac muscle, myocardium. Okay? So what does this mean? Why is this so significant that I'm repeating it and I'm making such a big deal about it? Well, what that means is that instead of having a depolarization and then immediately repolarizing, these L-type, remember, long-lasting. Okay, so this would be like a skeletal muscle. It would just depolarize and then repolarize again, unless you kept the signal there. Cardiac muscle doesn't do that. Cardiac muscle, remember, sodium rushes in. That's what happens during an action potential. That's what this little thing was showing. It's showing depolarization, sodium rushing in, depolarizing it all the way down. So that's the same. Sodium rushes into the cardiac muscle cell, and that's fine. And it's supposed to be, remember, immediately after that, potassium rushes out. That's an action potential. An action potential is very quick in the muscle and in the nerve. In the muscle and in a neuron. Okay, so it's supposed to be that fast. The problem is, and the difference is, this calcium. Okay, so instead of potassium, it's not permeable to potassium right away. That's what this P means, permeability to potassium. It's not permeable to potassium right away, so it doesn't simply depolarize immediately. It stays, or repolarize, I'm sorry. It doesn't repolarize immediately. It stays depolarized because of this increase in the permeability to calcium. And so it depolarizes, stays depolarized for a little while, and then repolarizes. Okay. So do you remember talking about refractory periods? Okay, and the refractory period, I think I probably made the analogy of like flushing a toilet. You flush the toilet, and then for a little while the handle just jiggles, and you're not able to get it to flush again because if it's a refractory period, it's already flushing. And so in the same way, when a muscle or a neuron depolarizes, as long as it's depolarized, you can't depolarize it again for a little while. Okay, now try to make sense out of this. The way that we can sustain a muscle contraction is that we can keep depolarizing it. So it depolarizes, and the muscle, here's what the muscle is doing. The muscle is actually contracting here. The muscle contracts a little bit more slowly. Okay. And so what we can do is if we could depolarize it quickly and then depolarize it again before the muscle is done contracting, are you following me? Then this muscle will contract again. And then if we could do that again, here, the muscle will contract again. And we did that. That's part of what our labs were. And we just keep contracting the muscle. That's what we can do with skeletal muscle. And we can have a long, sustained contraction with the skeletal muscle. You can keep your skeletal muscle contracted for as long as you have enough ATP and calcium to keep it contracted, okay, until you fatigue out. Guess what? You can't do that with a cardiac muscle, which is good, because why would you want to? Why would you want your heart to contract and then stay contracted? So what happens is that when you tell your cardiac muscle, your myocardium, to depolarize or to contract, it contracts, and then there's no more signal you can give it because it's still depolarized. Okay? That's very important. And that's because it's permeable to calcium. The calcium is still rushing in, keeping it depolarized, and so it can't depolarize again until a complete contraction has taken place. I hope that makes sense. Again, this is cardiac muscle. That's the way it works. With skeletal muscle, you could just keep depolarizing it until you maxed it out, and then it would just stay depolarized. That's the difference. Skeletal muscle can do this because you can just depolarize it here, and then you can depolarize it again, and depolarize it again. And look at all the numbers of depolarizations, probably faster than this, that you can do in the same amount of time that you get one depolarization event for cardiac muscle. That is the biggest difference, and it's so important because you don't want your heart to just beat or contract and stay contracted. Okay, you don't want it to be able to do that. You want it to contract, release, because it's a pump. Okay, you don't want any of this. Okay, no summation. That's what we call that, summation. Okay, we don't want that. All right, so here's another picture of this because it is an important concept. And so you have this depolarization event, and then you have calcium inflow. So the calcium is coming in, keeping it depolarized, 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 depolarized. And this is the term we use. It increases or it lengthens the refractory period. Okay? So during that time, no other signal can come in and cause a depolarization. And while all that's going on, this is the electrical stuff. That's what this graph is showing, the electrical stuff that's going on. During that time, the muscle itself is going, okay, I'm depolarizing or I'm contracting, and now I'm releasing. Okay, so that happens in about 0.3 seconds. So then about 0.3 seconds later, it's ready to go again. The refractory period is over, and it can beat again. Now, 0.3 seconds is about just under a third of a second. Okay, so that means that it's a little bit less than that, so maybe a fourth of a second. Okay, try to follow this because it's actually very interesting if you understand that. Let's say so from here to here is 0.3. Let's say that 0.25 is this point here. So that means that's about 1/4 of a second. Okay, you agree with me on that, right? So from this point to this point, this refractory period, if this is that much, we'll just say, just for fun, this is about 0.25 or a fourth of a second. So that means that the muscle contraction takes place, and then it's ready to go again right here. So that means now you can contract the heart muscle again, which means that if one contraction takes about a fourth of a second, that means the best you can do for contracting a heart is about 4 times per second. It's going to be different in everybody, but this example kind of works that way, which means that if you're talking about minutes, that means the maximum number of contractions you could possibly have is 60 times 4, which is 240 beats per minute. So that's a maximum. Physiology says, nope, you cannot contract, you cannot beat a heart faster than, if this is to hold true, if this is a fourth of a second, then faster than 240 beats per minute. That's it. That's your max. And usually our max is a little bit lower than that, but that's the maximum heart rate. Usually it's quite a bit lower. It's usually in the low 200s or something like that. But for this example, it works because it gets the point across. Okay. Now, okay, so let's do another, just because it is important, let's do another comparison. This is skeletal muscle. This is what I was trying to draw earlier. This is cardiac muscle. Skeletal muscle, you have a depolarization. It's the same thing. And then your muscle contracts. Okay? So because the muscle contraction takes a little while, it doesn't happen as fast as a depolarization event does. That's just like a snap, sodium out or sodium in, potassium out, done. Okay, the muscle contraction itself, because you have to go through bind troponin, tropomyosin, myosin walks along the actin, all that stuff takes a little while. Okay, so what that means for skeletal muscle is you're ready for another one right here. There's your refractory period. This is your refractory period right here. Okay, so that means that I'm good to go for another contraction. Okay, I could just keep sending that signal, and this thing would never repolarize. I could send another one here. It would just keep going, and eventually it would just max out. Look again, try to understand this. Here, depolarization, calcium. Calcium leaks in, keeps it depolarized. That is your refractory period from here to here. Again, about a fourth of a second. Okay, so the muscle contracts, and then you have almost complete relaxation before you're ready for another contraction. Okay? Okay, so hopefully that makes sense, and you can see this difference, because the difference between that refractory period and this refractory period is the biggest difference between skeletal muscle and cardiac muscle, and why cardiac muscle cannot stay contracted. You can't use it in the same way. You can't pick up something heavy with your heart because you can't keep it contracted. Okay, so let's kind of put those things together. You have your SA node, so the electrical stuff. You have your SA node that sits up here. It causes a depolarization. Okay, so what that does is it spreads out to the atria, all through the atria, and all of these muscle cells will depolarize at the same time. Okay, so then you have complete depolarization of the atria. We call that systole, or we call the contraction part of that systole. And then you have a pause. You have a pause right here at the SA node, where the SA node moves into, so that you can see it a little better. That's the I'm sorry, this is the SA node that sits up here, that was activated initially. This is the AV node. Okay, so that's where you have the little pause. Okay, depolarization spreads more slowly. Conduction slows through the AV node. Okay, so it slows, and then it moves down through the left and right bundle branches of the AV bundle of His, spreads down to the Purkinje fibers, and then to all of the ventricular myocardia, or the ventricular heart muscle cells. And then you have contraction of the ventricles. Contraction of the ventricles is something we call ventricular systole. Okay? All right, so hopefully that makes sense. All right, so all of this depolarization and repolarization are electrical. Okay, I'm going to write that because it's important. I'm going to try to spell it right though. Electrical. Okay, electrochemical actually, because we're still talking about that same thing we've been talking about since the very beginning, and that is sodium rushing in and then potassium rushing out. Okay, depolarize. Potassium rushing out, repolarize. We've been talking about that since we first started talking about cells. Okay, so we have a way to measure that, and we can actually just put little electrodes on the surface of the body, and we can pick up on these electrical changes, and that's what an EKG does. It's measuring the depolarization and repolarization of all of the myocardia. Not, you can see it, not the autorhythmic cells. Okay, that's going to be what I'm going to be accused of having as a trick question. But here's your answer right here. It's not the autorhythmic cells. It's the myocardia. Okay? So there, it's not a trick question because there's the answer right there. So your EKG, all of these little things are showing the myocardia. All of these muscle cells, remember, they all work as a unit. They all depolarize at the same time. They all repolarize at the same time, or about the same time. And that's the electrical signal that you're seeing. The autorhythmic cells are just a small group of cells. We can barely see it. Okay. Now, if you have a very sensitive EKG, you might be able to pick it out, but that's not what we see when we see an EKG signal that looks like this. Okay. We'll get into what that is. Okay. So, let's see, if the conducting pathway is normal, then this is what you see. If it's abnormal, you start to see little differences. Okay, and we're going to talk about what some of those differences are in this EKG signal. But if the pathway is normal, if the heart's enlarged, if parts of the hearts are damaged, then all of these things look different. And so we have recognizable waves. There's the P, okay? And what this is depolarization of the atria, okay? So it's a smaller signal. The atria are a little bit smaller, but guess what this big signal is? If this is depolarization of the atria, remember Depolarization means that's sodium rushing in. If that's depolarization of the atria, then this signal must be depolarization of the ventricles, because the ventricles are larger. Ventricles. And that's surely what it is. It's depolarization of the ventricles, and we call that the QRS complex. Okay, QRS complex. Now we have repolarization, and if depolarization of the atria was not too big, well, guess what? Repolarization happens right there, and it's about this big, and you can't see it. You can't see repolarization of the atria. You can, however, see repolarization of the ventricles. And that's what the T wave is right here. Okay, so you got that? The P wave is depolarization of the atria, so your little autorhythmic cell told all the atria to depolarize at the same time. We have leads that are sitting on the body, and they say, oh, there was something, and that's depolarization of the atria. And then those atria repolarize, but it's so tiny we can't see it. And besides, there's depolarization of the ventricles that's happening at the same time, and that's a huge signal. So that's what you see. You don't see that repolarization of the atria. Depolarization of the ventricles, we call that the QRS complex. There's a lot of electrical movement going on there, so it actually has these two little shoulders, or these little dips that occur on either side. And then repolarization of the ventricles is that T wave. Okay, so when we look at this, and I mean, somebody's going to listening right now is probably going to end up doing something with the heart, and so usually we use like a 12 lead system, and we put leads all over the heart. Now this is a 3 lead system. We put it on the chest above the heart. Um, this is a 3 lead system, and basically what it's doing is it's measuring across this way, it's measuring across this way, and it's measuring across this way. So you're getting a few different views of what's going on with the heart, but mainly it's lead 2. Okay. Um, uh, that doesn't say lead 2, but usually it's lead 2. That's the one we're actually looking at, and it's measuring the direction of flow. Okay, so it's measuring which direction this depolarization is taking place. Okay, and so that's what we're getting. Okay, depolarization of the atria, then depolarization of the ventricles, and then repolarization of the ventricles. Okay, so P, Q, R. Yes, I know I'm repeating myself. QRS and T. Okay, so that's basically what we're doing, and we can get even more information. We can look at this from different directions. We can get even more information by putting leads all over the chest to actually try to get different perspectives, and that way we can really focus in by comparing a couple of different leads. If we have abnormalities, we can actually focus in and say, oh, there is definitely something wrong right here in this area, or we could focus in and say there's something wrong in this area. Okay, but let's forget about that, and that'll be, if you actually go into this, you'll learn a lot more details than we have time for now. Okay, so different waveforms. This is review. Okay, I know, review from what I just talked about. The P wave, small upward deflection right there, represents atrial depolarization. I already said that. Spreads from the SA node throughout the atria, and of course it comes down to the AV node where it pauses, and then you have the QRS complex, which is this right here, this entire complex right there, and that is a large upright triangular wave flanked by 2 smaller downward waves, these guys, and that represents rapid ventricular depolarization. Now, something to notice. I'm not saying contraction. I'm saying depolarization, because an EKG is measuring electrical information. Okay? And then there's the T wave. We've already mentioned it. That is repolarization of the ventricles. Okay? And remember, this is the other trick question. Where does repolarization of the atria take place? Well, it takes place right here. And there's a question that you're going to be asked, and you can either put that it happens or you can put that it doesn't. But that's where it takes place. It takes place at the same place as the QRS, or ventricular depolarization, and so we really don't see it. but it's there. We'll just never be able to pick it up because the other signal is so big. Okay, so this is the same thing. Repetition again. Depolarization of the atrial produces the P wave. Okay, and then, and this is kind of new, adding to that, we have this event called contraction or systole, and that's how you say that. Oops. Systole. Okay, so that's systolic. Okay, so hear this. Depolarization happens first, right? Because it has to depolarize, and then you have contraction. So systole follows, or the contraction follows the depolarization, and it's going to be like that all the way through this cycle. So we have the P wave of atrial depolarization, and then contraction, or atrial systole. Then you have depolarization of the ventricles, followed by, very, very quickly followed by, deep systole, ventricular systole, or contraction of the ventricles. And then, following that, you have repolarization of the ventricles, which then causes relaxation or diastole. Okay. Now we left out atrial diastole because it happens at the same time as ventricular systole, or the repolarization happens at the same time as ventricular systole. Okay. Um, all right. So let's look at some of these, some of the electrical activity. Here's just a bunch of normal stuff. If we say this one is normal, then each of these is about 0.2 seconds, 0.0, 0.04. So each of these has a set, and we're not going to get into it, and you can't really see it anyway. But you can see that the timing of these is just where it's supposed to be. So that's a normal EKG. We have the P wave, the QRS, and then the T. Okay, so this is considered normal. Now, if we look at that and we compare it to this one right here, what we see is we see the P wave. The atria are depolarizing very, very rhythmically. That means our SA node is functioning just fine. It's communicating with the atria, and the atria are depolarizing. The problem is that what's supposed to happen after the atria depolarize? You're supposed to have ventricular depolarization. Well, look, these ventricles, they're kind of doing their own thing. Okay, so that means in the heart, okay, so if we have the atria up here, and then the ventricles down here, remember we have the SA node that's trying to communicate, going through the AV bundle and then to the Purkinje fibers. Well, guess what? This person probably had a heart attack and damaged this area of the heart. So here's the AV node, or the SA node, saying depolarize, depolarize, and they're doing it every time. It's working just fine. But then when it tries to send that signal, because remember I said this is a very important point, the only way that that signal can get through to the ventricles is through this area. And if you have a block, if this is damaged, then you have no way to get that signal through there. Okay? And so that's what this is showing. It's showing a third degree block. Okay? So that means that this is completely, very rhythmic, but completely independent, and you're depending on the autorhythmic properties of the ventricles to even make this happen. Okay? So you might be able to stay alive for a little while with something like this, but you're going to have that fixed very, 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 very quickly. Okay? All right, so then let's look at a couple of more. This one, you can see that the ventricles are being very rhythmic, more or less, but the atria, well, I guess they're not that rhythmic, but the atria aren't doing what they're supposed to be doing at all. Okay, so they're sending signals down, and the ventricles are picking them up every now and then. So you have ventricular contractions, but the atria themselves aren't showing. You don't have any P wave at all. Okay. Okay, you can kind of see a little bit of a T wave, so you can kind of see that the ventricle is depolarizing and then trying to repolarize, but there's really no sign of, if I erase all this, there's really no sign of a P wave. So that means that the atria is fibrillating, which means that that means that they aren't beating on beat, okay? They aren't contracting rhythmically the way they're supposed to. Part of it is depolarizing, so if we look up here, part of it may be depolarizing, contracting, and then other parts of it, and so it's just kind of sitting there vibrating. Well, we can get away with that, right? Because it's atria, okay? And the ventricles are still filling, they're still getting blood, they're still going, so, that's not as life-threatening. It's not something you want to live with, but it's not life-threatening. This is a problem. Okay, down here, ventricular fibrillation. The ventricles aren't really doing anything. They're just all kind of vibrating, contracting whenever. There's really no rhythm at all. This is a problem, okay? Because you're not getting good blood flow through the body, which is the real issue. You're also not getting blood flow through to the lungs. Your blood is not flowing the way it's supposed to flow. You can't last very long with something like this going on. Okay, so this is where you would have to shock it, and you would have to reset everything. Everybody depolarizes at the same time, and then the SA node, and that's what happens when you put the little paddles on. Of course, you're supposed to put the paddles on here and here, and then you give that shock. Okay, a defibrillator. You give a shock, everybody depolarizes at the same time because they're forced to, because you've given the shock. There are your little paddles. Clear. You shock everything, and then the SA node is going to be the first one to recover from that, and it's going to say, okay, I've got this now, and it starts to send out the signal like it's supposed to. That's the idea anyway. Okay, so then if we look at this one, it says to analyze it. I guess we could analyze it. But you can kind of see that sometimes, so there's your P wave. Your P wave is happening, pretty rhythmically. And you can also see that the QRS is following it sometimes. Okay, so it followed it here, followed it here. So every time the P wave goes, it's following, or I'm sorry, every time the QRS complex goes, it's following the P wave, but it's not doing it all the time, which probably means, if we go back up here, oops, erased too much. That's our AV bundle. What it probably means is we have a partial block. Which means that it's trying to send the signal down, but every now and then that signal doesn't really get to where it's supposed to be going. So that's a partial block. This is a third degree. This might be a first degree block, which means that it's working every now and then, but you've definitely got a problem. Now, somebody can be up walking around and living with something like this for a while, but that's definitely something that needs to be fixed. Okay. And that really comes from the scar tissue, okay, because that's where we say change in PQ interval. That's another thing that we might see. In this case, it's actually missing, so this might be, but this is also a heart block down here. Okay. All right. So that gets us to one of the last things we're going to be talking about, and that's the cardiac cycle. Okay. So the cardiac cycle means all events associated with a complete contraction. That means the atria contract, so atrial systole, just contraction of the atria, atrial diastole, relaxation of the atria, ventricular systole, contraction of the ventricle, and ventricular diastole. Okay, so that's the contraction part of the event. So I said this earlier. So if we focus on atrial systole, that means the atria are going to contract. Remember, they were told to do it by the SA node, usually. So depolarization of the atria follows depolarization of the SA node, forces blood, and this contraction, or the systole is contraction, will follow that. Okay, and that will force the blood from the atria into the ventricles. Okay, and we already mentioned the slowing that takes place here. And then it will relax. Okay, so the atria repolarizes, and after the atria repolarizes, it relaxes. Okay. And we also said that it's masked by ventricular systole. Okay, then we have ventricular systole, which happens after depolarization. So remember the QRS, and then we have systole, that takes place. Okay, so as soon as that depolarization happens, we have the muscle contraction that takes place of the ventricles. Okay, and then ventricular diastole. So that means that you have repolarization, and that's when the muscle relaxes. Okay, I didn't draw this very well, but if we were to put it all together, depolarization, the atria, depolarization of the ventricles, repolarization of the ventricles, then what we would see is the atria would contract here, and then the ventricles would contract, and then they would relax as the repolarization took place. Okay? All right. I have a picture that's going to put all of this together called the Wiggers diagram that's coming up. Okay, so a couple of terms that you need to know, and these are just terms with a little bit of calculation idea. Cardiac output. So one of the things we're interested in is how well your heart is working. And so we want to know how much blood it's pumping because that's really its job. So the more blood it's pumping or the better it pumps blood, the better or healthier your heart is. And so we have a way of measuring this. It's called cardiac output. And what it does is it takes your heart rate. Okay, that makes sense. How many times it's beating per minute. And then it's saying, okay, with every beat, how much blood is coming out. Okay, and we call that stroke volume. And all you have to do is multiply these together and you get cardiac output. Okay, so let's go over that. So the heart must increase or decrease based on metabolic need. Okay, so if you're running, your heart's beating faster. Okay, cardiac output is the volume of blood ejected from the left or right ventricles into the aorta or pulmonary trunk each minute. Why is it each minute? Because it's depending on heart rate. Okay, very important. Cardiac output is each minute. Okay, so we have this little thing called stroke volume, which is very simply how much blood is being ejected by the ventricle during each contraction. Okay, so then we have everything we need. It's very simple, actually. Cardiac output, you just take that stroke volume, how much blood's coming out, times how many times it's beating in a minute. Okay, so it's like if you want to figure out, how much water you're pouring into a pitcher, you figure out, how many cups, and you figure out how fast you're putting it in. Okay, that might not have helped you. Okay, so stroke volume times heart rate. So that's what it is. It comes down to this calculation right here. Okay, so then you just have to know what all of these things mean. And so if we calculate this, an adult male, let's say every time the ventricle squeezes, it squeezes out 70 milliliters of blood, and it does it, well, let's see, we don't need that per minute, and it does it 75 times per minute. Well, when you multiply those together, this would be 0.07 milliliters, then you end up with 5.25 liters per minute, and that's actually pretty average. Okay, now the blood volume of an average man is probably about 4 to 5 liters. Okay, so if we figure 5 liters of blood are in this person, then we can see that every minute he is pumping at least, this says more than 5.25, more than his blood volume, but it's around the same every minute. And that's probably what you're doing, is your own blood volume every minute is being pumped through your body. Okay, and then one more thing, cardiac reserve. So we know that when we run or jump or get scared or something like that, our heart starts beating faster and starts beating harder. Okay, that's very important because we want to have that reserve capacity. Okay, we don't want to be just sitting there relaxed on the sofa pumping with our heart beating and pumping as much blood as possible, because that's the most we could ever strain ourselves. Okay, so we have this maximum cardiac output minus what we're doing when we're just sitting there. So how hard can my heart beat versus how hard does it beat when I'm just kind of sitting around? Okay, and that's our cardiac reserve. Okay, so usually, so you right now, if you're sitting here listening to this and you're not doing it while you're on a treadmill, but if you're relaxed, you can actually pump about 4 to 5 times as much blood per minute. So your cardiac output could be about 4 to 5 times more than what it is right now. Okay? So if you have a if you have serious heart disease, you may have very little or no reserve, and so your heart may be beating as much as it can at any one time. Um, okay, and stroke volume. And the reason this is able to happen, one of the reasons you have so much cardiac reserve is because when you're sitting there and you're not doing anything, your ventricles aren't even squeezing out all the blood that they're getting. They're only squeezing out a portion. They're just squeezing out a little bit of blood, about half the blood that they have. So think about that. There's your, let's see, your atria, your ventricles are down here, and they get a certain amount of blood. Okay, so that's the amount of blood that they get. And then when they squeeze it to the body, they're only squeezing out about half of it. They're actually keeping some. So that's the reserve. So all you have to do is increase the heart rate and extract all that, because you can, that stroke volume, that's what that is, can be 100%. You can squeeze all of that blood out if you absolutely have to. Okay, so this is the diagram. This is in your book. And this puts everything that we've been talking about together, and then some. Okay, so this is our EKG. So we can see that if we just follow it through for one of these, we can see we have atrial depolarization, and then we have contraction. Okay, so then let's see, left atrial pressure, we can see that the pressure in the atria increases. This is the green. We can see the left atrial pressure increases. Okay, because after depolarization, we have contraction. Okay, and then there's a little jump here because as the ventricle starts to squeeze, it actually causes some back pressure. And then, so now let's look at the ventricle. So this is the ventricle, this blue one right here. Sorry, I'm marking the blue one with red. But you can see as the QRS complex, you have depolarization, and then immediately after that, the pressure in the left ventricle increases and goes way up. Okay, so this is showing your pressure goes way up to like 120. Oh, that's like blood pressure. Okay, that's exactly what causes blood pressure, is that ventricle squeezing. Okay, so we've already talked about the sounds as well. That's on here. So when the AV valves, or the mitral valve and the tricuspid valve, slam shut after the blood moves in and the ventricle starts to squeeze, it squeezes that AV valve shut, then you have that turbulent flow and you have the sound 1 and then sound 2, and then they fill up again and you have sound 3 and 4. That we're not worried about those right now. Okay, and then you can see that the ventricle is emptying. So there's a lot of information here, but hopefully it can make sense. I'm not going to quiz you per se on something like this. There's a lot of stuff that we didn't talk about, but in general, if you look at, and this is something I do want to draw your attention to. If you look at the aortic pressure, the aorta, when the ventricle squeezes, okay, right here, it's the QRS, that's the depolarization, and then right after that, the ventricle contracts, squeezes blood into the aorta, and the aorta says, whoa, I'm getting a lot of blood. And then, as the ventricle relaxes, the aorta starts to lose some of that pressure, but then that semilunar valve slams shut, and so that aorta keeps the blood in there. Okay, the blood doesn't have anywhere else to go except forward. And so there's constant pressure inside your blood vessels. And we're going to talk about that later. There's constant pressure inside your blood vessels. It never goes down to zero. They never empty completely, which is essentially what would have to happen for them to go to zero. Okay, so they're always running between this range. So if you look at the aortic pressure, it's always between about, let's see, this is about 78, something like that. Maybe we could say 75 and maybe 115, something like that. Okay, and this is exactly when we're measuring systolic pressure and diastolic pressure. That's exactly what we're seeing. Okay, so if that doesn't make sense completely, we're going to hit it again, so it might. Okay, and then this shows pictures of everything that's taking place. Okay, sorry so long. Good luck with it.